Is the screen is visible? The screen is visible?
Yes, ma'am.
Yes,
Okay.
ma'am.
Okay, so today the topic is molecular basis of inheritance. Okay. So what is molecule? So commonly what is called as molecular? Can anyone say? Can anyone say what is molecular? Molecular. No idea? You haven't read this topic before? Answer. Nandini, Ajit Kumar, Leah. Answer me, da. Whether you have studied or not. So according to that,
We have.
we have. Then why you didn't people didn't ask answer my question? Okay, no problem. So we'll see the molecular basis of inheritance. Okay, that we are going to see about DNA. So all the genetic materials we will be seeing. Okay. So molecular inheritance. Okay. So that is mean by it is DNA or RNA. They are totally called as genetic materials. So what is the role of genetic materials? What is the role of genetic material? No one knows. What is the role of genetic material?
It carries the genetic information from the parent to the progeny.
Yes. What about others? Okay. So what are the four requirements of genetic material? Okay. So it must carry information. So you already told, no? So it must carry information from one generation to another generation. So that is the main property of genetic material. Whether it is DNA or RNA, the main property, the first property, it should carry information from one generation to another. Okay. Then what are the other properties? They include replication. So do you know about DNA replication, RNA replication? Have you heard about it? DNA replication? Replication of DNA? No one? Okay, so DNA should replicate itself by itself. Okay, should replicate, should self-replicate. Okay, so it should have the another second property is that it should self-replicate. Okay, then the third property is that it must allow information to change, that is mutation. Okay, what is mutation? See, so this is the structure of DNA. Okay, if there is any change in the structure or any change in the property, okay, it leads to a concept, a phenomenon called mutation. Okay, there are many types of mutation. Okay, physical mutation, chemical mutation. Okay, so these mutations will cause some disorders. Okay, there are this leads to genetic disorders. So can anyone name any two genetic disorder? Anyone can say? Name any two genetic disease. Nandini, Ajit Kumar.
Ma'am, Down syndrome, Patas syndrome.
Okay, fine. Down syndrome. So it's a very good example for genetic disease. Then what are the other disease that you know? Alzheimer disease. Okay, so many diseases are there based on the genetic characters. So it will be classified as like based on the structural modification, based on the chemical modification. Okay, so it differs. Okay, so some change in genetic information leads to mutation. Then fourth point will be the gene. It should express gene. So each gene has its own property. Okay, so each gene must express its own function. So expression should be there. So one another more important point, important property. Okay. Okay, next. So we'll see the structure of DNA. So you people will know this already. But we'll see it again. Okay. Okay. So this is the structure of DNA. So the parts, the components include minor groove. Groove means some bend. It's like a bend. Okay. So bend, gap. So it can be that sort of meaning. Okay. Minor groove. Minor means small. Major groove, large. Okay. So the gap or the turn will be more. The gap will be more. That is called major groove. Next. So this gray color is called as sugar phosphate backbones. This gray color is called as the outer covering. Okay, the border. So that is called as sugar phosphate backbones. Okay. Then inside we have the adenine, thymine, guanine, cytosine. You know that, right? So those are called as base pairs. Okay, we'll see which are all adenine, which are all coming under purine, and which are all coming under pyrimidines. Okay, we'll see that. Okay. Okay, see here. So it is a right-handed helix. So the turn will be like a clockwise. Okay, so it is called as right-handed helix. Okay. Next, then between nucleotides, nucleotides, the adenine, guanine, those are called nucleotides. Okay. So between the nucleotides, the space will be 0.33 nanometer. For each nucleotide, the space, the gap will be 0.33. Okay. Next, for each turn, so this is a turn, right? Okay, this turn. Okay, for each turn, there will be 10.4 nucleotides. Approximately 10 nucleotides will be there for each turn. Okay. Then it is made up of two grooves, my major and minor grooves. Okay. Okay. So these are the points regarding the structure of DNA. Okay, we'll see two factors called nucleotides, nucleoside. Okay, so what are nucleoside and what is called nucleotide? Okay, so nucleotide is combination of sugar, some ribose sugar, deoxyribose sugar. Deoxyribose sugar is present in DNA. Ribose sugar is present in where?
RNA.
RNA. Okay. So in case of DNA, a sugar along with nitrogenous base is called as nucleoside. Okay. Sugar, it may be deoxyribose sugar that is in DNA or just ribose sugar in RNA. Okay. So this nitrogenous base and the sugar compound are combined with glycosidic bond. Okay, nitrogenous base and sugar combines with with the help of glycosidic bond. Okay. So in other words, nucleoside is the combination of sugar and nitrogen base. What about nucleotide? Okay, nucleotide is nothing but sugar, nitrogenous base plus phosphate. Okay, it's nothing but just one additional compound present in nucleotide. In case of nucleoside, only two things are there, that is sugar, one sugar and nitrogen base. Then in case of nucleotide, sugar, nitrogenous base along with phosphate is present. See this diagram? Phosphate, sugar, okay, ribose and glycosidic bond is attached between it, then nitrogenous base. Okay, you should not confuse with nucleotide and nucleoside. Nucleoside has only two. Okay, sugar and nitrogen. Nucleotide it has three. Okay, sugar, nitrogenous base and phosphate. Got it?
Yes, ma'am.
You can make note of it, okay, so that you won't forget. So many students will make mistake regarding this nucleotide and nucleoside. Okay, they will get confused with both. So you be clear. Nucleoside is just two components, sugar and nitrogenous base. Nucleotide is three components. You remember it as nucleotide T. T for 3. Okay, so it is made up of three components, sugar, nitrogenous base and phosphate. Okay. Next, purines and pyrimidines. So as I told already, so the base pairs are called adenine, guanine, cytosine and thymine. Okay. So in that, purines are adenine and guanine. Pyrimidines, cytosine, uracil, thymine. Okay, only two purines, A and G. Pyrimidines, C, U, T. In that, which is present in RNA? In pyrimidine, one pyrimidine is present only in RNA, not DNA. Which is that? Which base pair?
Uracil.
Uracil. So it is present only in RNA, and which is present in only in DNA? Yeah. Which is present only in DNA, whether it's cytosine or thymine. It's basic thing you must know. Nandini?
Question once more, ma'am.
So you told that uracil is present only in RNA. Yes, that's correct. So what about a base pair which is present only in DNA? Whether it's thymine or cytosine?
Thymine.
Sure?
Yes, ma'am.
Yes. For this answer you are thinking, what about Nandini and Leah? Okay. Okay, so there are two purines, A, G, that is adenine and guanine. Pyrimidines, there are three, C, U, T. Think it as cut. Okay, C, U, T. Cut or called as pyrimidines. C, cytosine, U, uracil, T, thymine. In that, uracil is present only in RNA. Thymine is present only in DNA. Okay. Next. So again, we are seeing the bonds, the presence of bonds in DNA. Okay. So between two base pairs, it is connected by the hydrogen bonds. Okay. The base pairs are linked by the hydrogen bonds. Then we have phosphate group here. See. Then sugar. So what sugar we have in DNA? Deoxyribose sugar. Okay. Okay. So these are called nucleotides. So as we already seen, right? Nucleotides have three components. That is sugar, phosphate group, base pairs. Okay. Nandini. Nandini, are you there?
Yes, ma'am.
Can you say what is nucleotide and nucleoside? Nucleotide and nucleoside. Don't know? If you are not saying the answer, you have to write your imposition. Is it okay? Leah Maria. Leah Maria,
Yes,
answer.
ma'am.
Yeah, answer it. So what is nucleotide and what is nucleoside?
Ma'am, nucleotide has a sugar molecule and with a phosphate group attached to it and a nitrogenous base, ma'am.
Okay. Okay, correct. What about nucleoside? Nucleoside?
Nucleoside is the same thing, but I think there's no phosphate group.
Why you think? Why you are not confirmed with your answer? So before 2 slides, I gave the explanation of nucleotide nucleoside. Nucleoside, which includes 2 components. Nucleotide, which includes 3 components. Nucleoside has sugar and nitrogenous base, only 2. In case of nucleotide, additional molecule is there, that is phosphate. Okay. Ajit Kumar,
Yes, ma'am.
can you say about purines and pyrimidines?
Yes, ma'am. Purine contains 2 components, adenine and guanine. Pyrimidine contains 3 components.
Okay.
Cytosine, thymine,
What
and
are those?
uracil. Cytosine, thymine, and
thymine,
uracil.
uracil. In that,
In that, uracil will be present only in RNA and thymine will be present only in DNA.
okay. Okay, that's all. Next, we'll move on to the next slide. Okay, so this is bDNA. So there are 3 forms of DNA, A, B, and Z. Okay, so this is bDNA, B form. It is right-handed helix. Okay, so as I already told, the turn will be in clockwise direction. So it is called as right-handed helix. Okay, then the diameter will be 2.37. Okay, so for each base pair, okay, base pair is adenine, guanine, okay, like this. For each base pair, okay, so for each base pair, so the gap will be 0.34. Okay. Then the distance per complete turn. For each turn like this. So the DNA will be like this, right? Okay. For each turn, the distance will be 3.4 nanometer. Okay. Number of base pairs for a single turn, there will be 10 base pairs. Okay. For a single turn, there will be 10 base pairs. Next, the major groove will be like wide, it will be large, okay? The minor groove will be narrow. So that we have already seen in the diagram, right? Okay, next, what are the other forms of DNA? B form, A, Z. In that, Z form will be the left-handed. The turn will be in anti-clockwise direction. So it is called as left-handed DNA. Okay, the diameter will be everything most are same, but except Z DNA, the diameter, helical diameter will be very small in compared to B and A. Okay, then the distance between each base pair is almost same, but in Z DNA it is a little much more. Okay, distance per complete turn. Okay, for each complete turn, what about the distance? That A and B is almost similar. In case of Z, it will increase. Number of base pairs per complete turn. So in already we have seen that in B DNA there will be 10 base pairs. In case of A, 11. Then Z DNA it contains 12 base pairs for each turn. Okay, so my major groove will be wide and deep. So that we have seen in a diagram. Then here narrow and deep. But in Z form, the major groove will be flat. Okay. So the structure of a Z form of DNA will be entirely different. It won't be like a normal DNA. Okay, in case of minor groove, the B DNA is narrow, and in case of A DNA it's broad, and in case of Z DNA the minor groove is narrow. Okay. Okay, so once again we are seeing the structure of DNA. Okay, hydrogen bond, sugar phosphate backbone is present. Major groove, minor groove, we have already seen. Okay. Okay. We'll see the experiment. So do you know this experiment, Griffith Experiment? Ajit
Yes,
Kumar?
Yes, ma'am.
So what for they are doing?
For this, to find whether the DNA is a genetic material or not.
Yes.
And it transfers, and it get transfers from one to another or not.
Okay. Okay. Okay, so you're correct. Okay. So what this person Griffith, his name is Griffith. Okay. So he first before many years. So the scientists don't know that some particles called DNA, it's responsible for transferring genetic material. So no one has any idea about it. So at first Griffith who discovered that DNA is a genetic material, but actually it's not proved by everyone. Okay, but he gave some concept regarding this. Okay, so what he did? So he took 4 mice. Okay, in that first mice, he injected, not added, he injected. The organism name will be Streptococcus pneumoniae. Okay, so this organism will cause a disease called pneumonia. So what kind of disease is that? Pneumonia? Can anyone know this? So it is a respiratory disease. Okay, it is a respiratory disease. Okay, it is caused by the bacteria called Streptococcus pneumoniae. So what he did is he took there are two types of strains. One is smooth strain and rough strain. So they are called as S and R. Okay, smooth strain is more infectious. Rough strain is less infectious. Okay. So what he did in the first mice, so he injected the rough strain of the Streptococcus pneumoniae. Okay. So after injecting, after some time, so the mice was alive. So it doesn't cause any infection. The mice was healthy. Okay. So when he injected S strain in the mice, so what happened is that the mice dies. Okay. The third step, what he did is he treated S strain. Okay. The normal S strain, that is a very infectious part. Okay. That infectious part, infectious strain has been he treated. Then after heat treating, that strain is injected into the mice. Okay, so after some time it was noted that the mice was alive. It doesn't cause any infection. So in the next step, what he did is he injected he treated strain plus S strain. He treated S strain plus R strain. Okay, so when he combines both heat treated and smooth strain and rough strain, but the smooth strain he didn't add it directly. Instead, he just treated with heat. Okay, then he mixed both strains and then he injected into the mice, and he noticed that the mice dies. Why? So what is the reason? Ajit Kumar? What is the reason?
Ma'am, some bacteria have been transformed from R to S, ma'am.
Yes, something, not bacteria, something has been. So he find that he concluded that something has been transferred from one cell to another cell, but he don't know what is that thing. Okay, so he couldn't give any furthermore information about it. So this concept has not been accepted by everyone. Okay, so this is Griffith Experiment. Okay, these are the steps. Smooth strain. So after adding smooth strain, the mice dies. Okay, rough strain, the mice lives. Okay, heat killed smooth strain, the mice lives. Then heat killed S strain and R strain, the mice dies. Okay. Okay, after that, three scientists, Avery, Macleod and Mechatic. Three scientists, they all combined to work with this Griffith experiment. So they need to find what is that. Okay, so some Griffith has told that he has concluded that something has been transferred from one cell to another. But these scientists have to confirm that what are those cells. So what are those things? They have to see. Okay, they are ready to study about it. So they did an experiment called Avery Macleod experiment. Okay, So this is the experiment. We'll see in this diagram. Okay. See, in the first test tube, so what they did is that they took a test tube. In that, they add some media for the growth of smooth cells. He added the heat. They added the heat-killed smooth cells of the Streptococcus pneumoniae. Okay. So this heat-treated smooth cells are used in Griffith experiment. The same cells are added inside this test tube. Okay. Then he added the rough cells. After the second step will be the addition of enzymes. Okay. So what he did is, in a test tube, he take the heat-killed S cells along with some media. So media means for food to bacteria. Okay, for that growth medium for the bacteria. Okay. So what he did is, he took 3 test tubes and he evenly distributed in 3 test tubes. Okay. Okay. He just evenly distributed in 3 test tubes. Then in that first test tube, what he did is he added an enzyme called protease. Okay. In second test tube, he added an enzyme called RNase. Okay. In third test tube, he add DNase. So these are called as enzymes: protease, RNase, DNase. So what is the role of protease is that it breaks down the proteins. It breaks down the proteins. So what is the role of RNase is that it splits, or it will break the RNA. Okay, the protein nature will be denatured here. Likewise, here after adding RNase, the RNA will be denatured. Denatured means it loses its property, then there won't be any substance. Like in case of protease, after adding protease, there won't be protein in this test tube. Okay. After adding RNase, there won't be any RNA present inside the test tube. In the last test tube, they added the DNase group enzyme. Okay. So after adding DNase, there won't be any DNA in that test tube. Okay. So this is the second step. First step, what he did is he took a long test tube and he added some heat-killed strains. Okay. Then after that, in the second step, he took 3 test tubes and he evenly distributed the heat-killed strains. In first test tube, he added protease. In the second, he added RNase. Okay, then third test tube, DNase is the enzyme added. Okay, so after adding, there won't be no protein here, no RNA here, no DNA here. Okay, after that, what they did is they add R cells. R cells is called as rough cells. We have seen in the before Griffith experiment, right? R cells, S cells. S cells means smooth cells. R cells means rough cells. Okay. So in the third step, they added R cells. Okay, so this is the R cell, this blue color. R cells, they added R cells in all the test tube. Okay, they transferred the substance into conical flask. Okay, to see the growth. Okay, so in that what they did is they add rough cells in all conical flask. Okay, then after some time they watch the presence of any growth. In first conical flask, the cells appear. The cells didn't die. Okay, in the second flask, the cells appear. So okay, it didn't die. Okay, in third conical flask, no S cells appeared. Okay, no cells appeared. So what it concludes is that no transformation occurs. Okay, so but in the first two conical flask, transformation has been occurred. In third flask, no transformation occurs. Okay. Okay. So that's why after this experiment, so the three scientists concluded that there are no growth in the last test tube which has no DNA. Okay, but in this conical flask, DNA is present. So in the second conical flask, DNA is present. But in the third conical flask, no DNA is present because we have added the enzyme called DNase. Okay, so they finally concluded that if there is no DNA, there is no transformation. So they give their observation as, so DNA is the responsible for the transformation process. Okay, understood? Ajit Kumar, Leah?
Yes, ma'am.
Understood? Can anyone say, or shall I repeat it? If you want to repeat, can. Ajit Kumar, Leah?
No need, ma'am.
Okay, do you understand? So then I will ask question after some time. Okay, so this experiment is Macleod experiment. You can say it as Avery and Macleod experiment. Okay, nothing is correct. Okay, so this is the second experiment. Then third one is Hershey and Chase experiment. Okay, so this experiment is done in 1952. So they did the experiment to confirm whether DNA is the genetic material. They just want to confirm it again. Actually, it is confirmed, but these these scientists, Hershey and Chase, want to confirm it again. Okay, so they did the experiment, the Hershey and Chase experiment, with the help of radioactive substance. Radioactive substance. Okay, we'll see in the picture. Okay. Okay, so what is this this structure? What is the structure? It's like a mosquito-like thing. What is this?
T for bacteriophage.
So what is the property? It is whether it's a bacteria or virus. So you told bacteriophage.
Ma'am, it's
Whether
a
it's
virus that
bacteria or
infect
virus.
bacteria, ma'am.
Very good. So it's a virus that infect bacteria. So that's why it's called as bacteriophage. Phage means eating, swallowing. Okay, bacteriophage, virus that eats or that digests, infect, any meaning is okay. Okay, so the phage or the virus that eats or the virus that infects bacteria are called bacteriophage. So the structure of bacteriophage is like this. Okay, so it has outer coat and this is DNA. It has DNA, genetic material inside it. Okay, so what they are doing is that, so they are using the bacteriophage. They have two experiments. They did the two stages. Okay, in first step, what they did is they labeled the bacteriophage with the help of radioactive sulfur. Radioactive, it's not normal sulfur, it's radioactive sulfur. So this radioactive sulfur coats only the protein coat, but not DNA. It won't enter DNA and it will label. It just label the protein coat outer. Okay, so that's why they have given like it's a fluorescent. So in that picture, they have given as a fluorescent light. Okay, so they added radioactive sulfur. Okay, in that radioactive sulfur containing media. Media means food. Okay, it's not like media, social media. So media means food for the growth of microorganisms. Okay, so they took some media, and in that media they added radioactive sulfur. After that, they allowed bacteriophage inside the culture, okay, inside the media. Okay, so what they observed is that sulfur, it coats only protein, but it doesn't coat the nucleic acid present inside it. So do you know that the bacteriophage won't enter completely inside the bacteria? Do you know that? So only the genetic material will enter. Wait. So this is the bacteriophage. Okay, this is the bacteriophage. This is the DNA inside it. So this bacteriophage will come, and the first step will be the attachment. So this is bacteria, okay? This is bacteria. This bacteriophage will come and sit over the cell wall of bacteria, and it won't enter completely. Okay, instead the genetic material which is present inside will enter. Okay, remember that the bacteriophage won't be entered, okay, inside the bacteria completely. Only it allows its genetic material to enter inside the bacteria. Okay, so it will just allow the DNA. Only the genetic material will enter. Okay, this is the life cycle of bacteriophage. Okay, that is lytic and lysogenic cycle. Okay, so here only the protein coat is labeled with the radioactive sulfur, and it is made to infected with some bacteria. So the whole protein coat will not enter. Only the DNA genetic material will enter, right? So after seeing this bacteriophage, there is no radioactive cells. Okay, there is no radioactive cells. So they after they performed one more experiment. Okay, they used some other radioactive substance, that is radioactive phosphorus. Okay, in first step they used radioactive sulfur. In the second step they used radioactive phosphorus, and this radioactive phosphorus will attach only to the DNA, will label or will combine only to DNA but not proteins. So it enters into DNA, okay, and it gets labeled, and the same step is followed. They allowed the bacteria to infect. Okay, then in the third step they have seen the radioactive DNA. Okay, so conclusion is the DNA is the genetic material. Okay, so they added two radioactive materials. One is sulfur, one is phosphorus. Sulfur has the nature only to combine with proteins, but phosphorus has their own nature to combine only with DNA. Okay, so after the infection with bacteria, they have seen no genetic material, there is no radioactive cells entered. But in the second experiment, they have seen some radioactive substance inside the bacteria. Okay, so they concluded that proteins are not genetic material, DNA is the genetic material. So by this experiment and the before experiment, okay, by these experiments only, the DNA is genetic material. So the concept is confirmed. Got it? Understood, Ajit and Leah?
Yes, ma'am.
Yes, ma'am.
Okay, next things are the basic common things, okay. DNA, which is the first was identified by the scientist called Meisher, 1869. He named he identified in first cells and he called it as nuclein. So they didn't name as DNA. First, the name of DNA was nuclein. Altman named it as nucleic acid. Altman, another scientist, he called it as nucleic acid. Then Watson and Crick gave the structure of DNA. Only these scientists only gave the structure of DNA. But Altman and Meisher, they didn't give the structure, but they just named. Okay, for Watson and Crick, they got Nobel Prize in 1962. Okay, DNA are seen in chromosomes of nucleus. So that you know, right? Okay, next thing is, so we have already seen purines, which include adenine, guanine, pyrimidines, which include thymine, cytosine, and uracil. Okay, nucleoside is base and sugar. Nucleotide is base, sugar, phosphate. Okay. Okay, so this is Chargaff's rule. In Chargaff's rule, the rule is concentration of purine will be equal to concentration of pyrimidine. That is, concentration of adenine will be equal to thymine. Concentration of guanine will be equal to cytosine. So, or it can be written as this form. A plus G is equal to T plus C. AG is equal to TC. That is plus. A plus G is equal to T plus C. Okay. So concentration of adenine will always be equal to the concentration of thymine. Likewise, concentration of guanine will be equal to concentration of cytosine. Purine and pyrimidine concentration will be the same. Okay. Examples, they have given an example in human, there are 30 adenine. So they are asking to find the percentage of other bases. Can you work it out? They have given the percentage of adenine. So can you say the percentage of other base pairs? Tell me the answer. Make it fast. Got it? Leah, Ajit, got it?
Yes, ma'am.
Answer?
Ma'am, adenine and thymine will be 30. Guanine and cytosine will be 20.
Yes. Okay, adenine is 30%. So the thymine will also be 30%. Okay. So they are everything combinedly to form 100%. Okay. So they are adding 30 plus 30, 60. Then 100 minus 60 is 40. Then 40 will be splits into 2. G is G and C are 20, 20. Okay. Okay, so likewise, there you will get some sums. Okay, bonds in DNA, hydrogen bonds. Hydrogen bonds and phosphodiester bonds are present in DNA. So that we have seen already in the diagram. So what is the charge of DNA? Negative charge. DNA is negative charge, okay? So there is double bond between A and T. A, T. And there is triple bond between G and C. Okay, so DNA is a negative charge between adenine and thymine. Okay, there is only double bond. Guanine and cytosine, there is triple bond. Okay, so DNA can be denatured by heating. So denaturation and renaturation are the two important properties. Okay. See, this is denaturation and renaturation. Okay, so this is the double helix. Okay, when it is treated with heat, okay, it binds. So the base pairs will be separated. So the word unwinds, unwinding. Okay, so if you are treating DNA with heat, it will unwind. When if you are giving more heat, the two strands will separate. Okay, but it can be renatured again by cooling. Okay, renaturation and denaturation and renaturation are the two important points of, they are the two properties of DNA. Okay. Okay, next is RNA. RNA as genetic material. Okay, so after that, some scientists came in conclusion that RNA can also be a genetic material. Okay, so RNA act as enzymes. So especially in transcription and translation, RNA will be acting as enzymes. So an experiment is done by Singer, Frankel, Conrad and Singer. Okay, they used tobacco mosaic virus for this experiment. We'll see the experiment. Okay, this is the picture. So what they did is, listen, this is one thing they have shown in purple color, lavender color, and one thing they have shown in blue color. Okay, so this is one type of TMV. Okay, TMV, right? Tobacco mosaic virus. So they took two forms of tobacco mosaic virus, two strains, TMV A and TMV B. What they did is, the second step, they did a degradation process. In that degradation process, these proteins will be removed from the RNA strand. See in the second picture. Okay, the proteins are removed. The proteins are like beads-like structure. Okay, proteins are removed from the RNA strand. That the spring-like structure is called RNA. Likewise, in TMVB, that is in blue color. Okay, same process is done. The proteins are removed from RNA strand. Okay, in the second step, what they did is, they exchange the proteins. The protein A is combined to RNA B. See, they have exchanged. Protein B is combined to RNA A. It's just shuffle, it's just exchanged. The second step, third step. After that, they are allowed to infect the leaf, tobacco leaf. Okay. So then after the infection, they, with the help of microscope, they view the cells. So if they are seeing, so what they observed is that what is progeny? Progeny is the offspring. Okay, it's a reproductive form of one microorganisms. Okay, so after infection there will be more number of virus here. So they are using the microscope, they view the cells. After viewing the cells, they found that both protein and RNA, okay, was the original form of TMV. Okay, see, they allowed to infect with the help of protein B and RNA A. Okay, but after the progeny was seen as the RNA, the color of RNA here used here. Okay, so likewise in the second type, they have seen the color of RNA, which is in blue color. Whole proteins are in the color of RNA. So what they concluded is that RNA is also the genetic material. Like DNA, RNA is also a genetic material. Okay, this is the experiment called Frankel-Condrat and Frankel-Singer experiment. It is done using tobacco mosaic virus. So what they did is they just remove the protein and RNA and they exchange the protein along with they exchange the protein of the two types. Okay, then they allowed them to infect with leaf. And after the infection, they have seen the cells as the original form of the type of TMV. Okay, so they finally concluded that RNA is the genetic material. Okay, so these are some points regarding to RNA. RNA is ribonucleic acid. They are single stranded. The sugar present in RNA is ribosugar. Okay, next tRNA, mRNA, rRNA are the important types. Okay, their main function is protein synthesis. So this is mRNA. So this is mRNA. Okay, this is tRNA. Okay. Okay, this is tRNA separately. It has 3 arms: D arm, T arm, anticodon arm, okay, extra arm. So this is called as amino acid acceptor arm. D arm, T arm, amino acid acceptor arm. Okay, these are the types of RNA, their structure and their function. tRNA will transfer amino acids during the site of protein synthesis. What is synthesis? Formation of protein. So during those process, tRNA will transfer amino acids. Next, ribosomal RNA. What is the role of ribosomal RNA? It combines proteins to form ribosomes. Okay, everything tRNA, rRNA, mRNA, 3 RNAs will include mainly in the process of protein synthesis. Okay, there are some other type of RNA, that is small nuclear RNA, micro RNA, then siRNA. So there are some other minor types. Okay, minor types which is involved in some gene expression process. Okay, siRNA is used in genetical studies. Okay, in research purpose. So they will prepare siRNA as an artificial siRNA and they will use it for genetical studies. Okay, so these are the difference between DNA and RNA. So the first thing is sugar, deoxyribose, ribosugar. Then another important difference is that DNA contains thymine, RNA contains uracil. Okay, So this is animal kingdom. One more topic we have to see. Okay, animal kingdom. So in case of animal kingdom, so they are collectively called as, just a minute. They are collectively called as metazoans. So what are metazoans? That is whether it is multicellular, unicellular. So based on those properties, animals have the properties like they are multicellular and they are eukaryotic. Okay, what is multicellular? They have more number of cells inside their body. Okay, so eukaryotic means they have proper nuclear structure. So what is heterotrophic animals? What is autotrophic and heterotrophic? Leah? Leah, are you there? Okay, so autotrophic and heterotrophic. Autotrophic means they can prepare their own food. Heterotrophic means it has to depends on other factors to prepare their food. Okay. Next. So basis of classification. Classification is based on their features like arrangement of cells, how it is arranged, then level of body organization, body symmetry, then whether it has coelom or not. Coelom means it's a hollow space, okay? Whether it patterns of digestive circulatory system or reproductive system. Based on all these characters, they are classified. Okay, we'll see the next topic. Levels of organization. Cellular level. Okay, so whether it is an unicellular or multicellular, based on that they are classified as unicellular, multicellular. Okay, tissue level. Okay, so based on their tissues, for example, coelant traits like Hydra. Okay, they have their distinct form of tissues. Okay, so they are classified according to their tissue level. Next, organ level. Okay, so based on the organ, some, for example, in case of respiratory system, we people have organ, lungs, nostrils for our respiration. In case of frog, fish, they have different organs for their respiration. Okay, so based on that, they can be classified. Okay, next, organ system level. Not only single organ, whole organ system may change in some living organisms. We'll see. Okay, the first thing is digestive system. That is incomplete and complete digestive system. Incomplete is that, so they have opening, so it acts both as mouth and anus. So in example for jellyfish, okay, those jellyfish will be having a single opening which will act as both the mouth and anus. For example, man, we have complete digestive system. Okay, starting from mouth to anus, we have complete digestive system. Okay. Same way, circulatory system, there are two types, open type and closed type. So open type, okay, so the open type, the circulatory system won't, that is blood won't flow in, especially like blood vessels, arteries, capillaries won't be present. So examples are arthropods, like arthropods include some insects, okay, some worms. Okay, so these arthropods won't be having separate heart, okay, or any arteries. Okay, they won't be having any arteries, vein, capillaries, okay, blood vessels. But in case of closed type circulatory system, they have arteries, veins, and capillaries. The example, the human. Okay. Okay, symmetry. So what is symmetry? There are three types of symmetry. One is asymmetry, asymmetrical. So in asymmetrical, the body cannot be divided into any halves, any two equal halves. Okay, in case of radial symmetry, see the picture. One is radial symmetry and one is bilateral symmetry. Bilateral symmetry can be divided into exact two halves. Radial symmetry can be divided, but not exact two halves. It can be divided into number of halves, number of parts. So this is radial and bilateral symmetry. So based on their embryonic structure, there are two types, diploblastic and triploblastic. Diplo means two. Okay, these embryo has two embryonic layers. One is ectoderm and endoderm. In case of triploblastic, They have 3 layers: ectoderm, mesoderm, endoderm. Okay. So based on the layers of the embryo, they are classified into 2: diploblastic and triploblastic. Okay. So this is the diagram which represents the layer of embryo. Okay. So this is the main classification for animals. So based on the kingdom is Animalia. Then based on the cellular level, unicellular, multicellular are there. Based on the tissue level, organ system level, it is divided into radial and bilateral. And when it is radial, there are two divisions: Coelenterata and Ctenophora. Then in case of bilateral symmetry, there are 3 types based on their cavity. Cavity means coelom. Coelom means cavity. Okay, body cavity. Next, coelom. So it's a cavity filled with fluid. Okay, fluid filled with fluid-filled cavity. So that are present in some animals. Okay. So what is the role of the coelom is that, so they help in flexibility. Okay, for the particular animal's body, for their movement. In that they have 3 types: acoelomates, pseudocoelomates, then eucocoelomates. Acoelomates means it don't have body cavity inside it. The body cavity is absent. Pseudo means false. So it has a coelom-like structure, but it is not the true coelom. So they are called as blastocoel. Another 3rd type is there, that is eucocoelomates. So there are 3 coelom types. Okay. So based on the classification, they are classified into 11 phyla. So we'll see it individually. First thing is Porifera. Porifera is the sponge which is present in marine environment. Okay. So they are multicellular organism. They lack nervous system. They don't have nervous system. They are non-motile. They can't move. Okay, there are holes and pores all over the body. So this is the first type, Porifera. Example is sponges. It is present only in marine environment. Next, coelentrates. Coelentrates. So these are the examples of coelentrates. Corals. And this is Hydra, sea anemone. Okay, coelentrates are radially symmetrical, which live only in water. Why? It can live only in water. Some are solitude. Solitude means It lives only in separate area. It won't mingle with others. But some are colonial. Colonial means group. Either they may live in separately, or they lonely, okay, or they will be living in as group. Example: hydra, sea anemone, corals. Next type will be the platyhelminthes. So they are mostly parasitic. Example will be liver fluke, blood fluke, tapeworm. So they are collectively called as platyhelminthes. Next, nematodes. So the body is cylindrical. Pseudocoelom is present. It won't have actually, it looks like it has a body cavity, but it don't have any body cavity. This type of animals, I mean nematode, will cause disease called elephantiasis. Elephantiasis is the disease, Yanikal Vyadi. So that is elephantiasis. Example: Ascaris. Next, annelids. So these are the annelids: earthworm, leech. Leech means atta. Okay. So these are the group of annelids. Okay. So they have true body cavity. They have body cavity that is cavity filled with fluids. Okay. They are found in variety of habitats. They don't live, or can live only in this area, or can live only in this area. So they don't have particular specific habitat. They can live in any habitat. Next, arthropoda. So it includes butterflies, prawns, okay. Circulatory system is open. They don't have arteries, blood vessels, okay. They don't have any those structures. They include some insects. Okay, these are the arthropoda, butterfly, insects. Most of the insects are called arthropoda. Okay. Next, mollusks. Okay, next one is mollusks. Mollusks are bilaterally symmetrical. Bilaterally means it can be divided into two halves. Okay, so they are triploblastic. Triploblastic means it has 3 layers of embryo. Okay, locomotion in mollusks is by the muscular foot. See, so see the snail. Then in case of octopus, then oyster, squid. Okay, so in all these animals, okay, the locomotion will be with the help of muscular foot. Okay. Okay, they have open circulatory system at kidney-like organs for excretion. Okay, they have open. What is open circulatory system? They don't have any blood vessels like our human. We have blood vessels which is connected to heart, and that is connected to lungs. Okay, we have closed circulatory system, but these group of animals will be having open circulatory system. The next one is Echinodermata. So the next group is Echinodermata. Okay, Echinodermata are triploblastic. What is triploblastic? Tri means 3. Blastic means embryo. 3 layers of embryo. Okay, with true coelomic. Coelom means cavity. Okay, so cavity. So it has cavity. So example is, see the picture, starfish and sea urchin. So these belongs to the group Echinodermata. Okay, they have spine. What is that? Spiny skin. Okay, like pointed skin. Okay, they have spiny skin. Okay, they are mostly spiny skin. The skeletons of echinoderms they have made up of hard calcium carbonate. Okay, their skeleton will be made up of hard calcium carbonate. So it's very hard. Okay, because a calcium derivative of calcium along with carbonate is present, the skeletons of these group of animals will be very hard. Okay, they exhibit radial symmetry. What is radial symmetry? So we cannot divide it into two halves, but we can see we can divide like this, but not exact two half. So it is called as radial symmetry. Next group will be the vertebrata. Vertebrata means it has perfect vertebral column. Okay. So in that we have many groups. We'll see that each one by one. First thing is Pisces, that is fish. Okay, they are mostly aquatic. Definitely they are aquatic, no doubt in it. Aquatic cold-blooded organism. Cold-blooded. So there are warm-blooded, cold-blooded. So why the name is denotes that it based on their living temperature. Because fishes always lives in water, so they are called as cold-blooded organism. Okay. So the body is covered by scales. So you have seen, right? The body of the fish will be covered by scales. Okay, then the muscular tail is for movement. So have you seen the swimming of the fish? So it will move with the help of its tail. So the tail is made up of muscle. Okay. So muscular tail. Next, amphibians. So amphibians are also called as cold-blooded animals. Amphibians can live both in, why they are called as amphibians? Amphi means both. They can live both on land and in water. Okay. So example is frog. Okay. So their respiration is by gulls and lungs. Gulls. Okay. Their heart is made up of 3 chambers. We have 4 chambers. Human have 4 chambered heart. But in case of amphibians, they have 3 chambered heart. And their reproduction is by eggs. Okay. So these are the points regarding to amphibians. Next, reptilia. So reptiles are cold, same again the cold-blooded. Most of them are cold-blooded. And their respiration is by lungs. Okay. Their heart is also 3 chambered. But except for crocodiles. Crocodiles, they don't have 3 chamber, they have 4 chambered. Okay. So heart of most reptiles are 3 chambered. They also lay eggs. Example, snakes, turtles, lizards, crocodiles. See the picture. Everything are reptiles. Alligators, snake,
Turtle.
crocodile. Yes.
Turtle.
Yeah, turtle, everything. So the animals which are in the picture or everything are reptiles. Okay, next is aves, birds. So birds are called warm-blooded animals. Okay, they are called as warm-blooded animals. And their 4 limb, we have like 4 limb, hind limb. Okay, every animals have 4 limb, hind limb. 4 limb is front. Okay, hind limb is back side. Okay, that the birds, the 4 limb of the birds is modified as wings. That is the purpose for flying. Okay, bones are hollow. Okay, hollow means it has some space. The bones will be made up of some space. Okay, the body is covered by feathers. We know that birds have feathers. Okay, respiration is by lungs. Okay, they have the 4 chambered heart like our human. Okay, so their reproduction is by eggs. So birds include every birds we know. Next, mammals. Okay, mammals are warm-blooded animals. Every mammals are warm-blooded animals. Okay, so mammals include all animals like lion, tiger, elephant. Okay, even bat. Okay, monkey, chimpanzee, we human. Okay, everything are called mammals. Why we are called as mammals? Because we give birth. We don't lay eggs. We give Birth. Mammals give birth. That's why they're called as, yes?
Viviparous.
Viviparous, yes. Okay, so we all mammals have 4-chambered heart. Their skin is covered by hair. Okay, all mammals will be, so like in birds, the skin is covered by feathers. In case of mammals, the skin will be covered with hair. Okay, hair. Then what are the other things which is present in skin? Sweat glands, oil glands, sebaceous glands. Oil gland is otherwise called as sebaceous gland. So we human have hair, sweat gland, sebaceous glands. So what are the purpose of these glands? To regulate the body temperature. Okay. So in order to regulate the body temperature, these glands and hair is present. Okay. Next, mammals give birth to young ones. We already seen it. Okay. So we'll have a recap. So we are seeing the animal kingdom. So animal kingdom, the animals, all animals are metazoans. So what is meant by metazoans is that they are multicellular, eukaryotic and heterotrophic animals. So they are called as metazoans. They are collectively called as metazoans. So what is multicellular? Can you answer?
More than one cell.
Yes. If the organism have more than one cell, it is called as multicellular. What is eukaryotic? Eukaryotic, you don't know?
Having nucleus structure.
Yes, having a proper nuclear structure. If it has proper nuclear structure, that is called eukaryotic. If there is no proper nuclear structure, then it is called as prokaryotic. Examples of prokaryotic is? Any idea? Which group of animal belongs to prokaryote? It's not an animal, actually, it's a organism, living organism. Which living organism? It's a prokaryote.
Algae.
No, bacteria. Only bacteria belongs to prokaryotes. Okay, rest of them, all fungi, algae, virus. Okay, then what else? Plants, animals, human, everyone are eukaryotes. Okay, then what is heterotrophic property? Autotroph and heterotroph. So autotrophs, they can prepare their own food. Heterotrophs, they don't prepare their own food, they depend on others. Okay, classification, based of classification, what we have seen. So the classification based on arrangement of cells, body organization, body symmetry, coelom, presence of coelom, then digestive, reproductive and circulatory system. The levels of organization is cell level, tissue level, organ level and organ system level. So in case of digestive system, there are two types, incomplete and complete digestive system. Incomplete is that they don't have separate opening for mouth and anus. They have a single opening which act as both mouth and anus. But in case of complete digestive system, they have two separate opening, mouth and anus. Circulatory system, there are two types, open system, closed system. In case of open system, So there are no arteries, blood vessels. Okay, so that is called open system. In closed system, arteries, vein, capillaries, blood vessels are seen. Okay, symmetry. In case of symmetry, there are three types: asymmetry, radial symmetry, and bilateral symmetry. Asymmetry is the body cannot be divided. Example, in case of sponges, we can't divide the parts into equal halves. Okay, but in case of radial symmetry, so the body can be cut into two equal halves. Okay, it can cut in any plane. Okay, so in case of bilateral symmetry, it can cut only through single plane. It can cut into two equal halves. Next, what about radial? Okay, have seen radial and bilateral? This is the picture for radial and bilateral. Diploblastic and triploblastic. So blast means embryo. Okay, diplo means two. Diploblastic, two layers of embryo. Ectoderm and endoderm. So which is present, only the two layers of embryo will be there. This diploblastic embryo will be seen in sponges. So triploblastic means three embryonic layers: ectoderm, endoderm, and mesoderm. Okay, which is seen in Platy helminthes. So these are the pictures of both embryonic structures, diploblastic and triploblastic. So this is the overall classification. This is the overall classification of animal kingdom. Okay. Okay, next, coelom. Coelom means cavity. Okay, cavity which is filled with fluid, some fluid. Okay, so what is the responsible of this coelom is that it provides some flexibility for the body when especially when it's moving. So based on that, we have three coeloms: acoelomates, pseudocoelomates, and eucoelomates. Acoelomates is that it has no coelom, no body cavity is present. Okay. So next, pseudocoelomate is that false coelom will be there. Okay. So that is it won't be like it will be like a coelom, but it's not actual coelom. Okay. Next, coelomates, eucoelomates is that they have true body cavity. So this is present in mollusca, annelidas, okay, insects. So true coelom will be present in insects, sponges, etc. In classification of animals, okay, there are 11 phyla. Okay, each phyla we have to see. Porifera, so we already seen. Porifera is that they lack nervous system. Okay, they have pores in it. See the picture. Sponge, example, sponges. These sponges have pores and holes in their body. Okay, they lack nervous system. Next, Coelenterata. Coelenterata is it includes corals, sea anemone. Okay, so what is that? We can divide into radial symmetry. Okay, so they live either single or in a group. Okay. Next, Platy helminthes. It includes tapeworm, liver fluke, blood fluke. Okay, so they are mostly parasitic. They won't live separately. They will depend on other hosts for their food and shelter. Next, nematode. It has no true coelom. Pseudocoelom is present. The body is symmetrical. Sorry, body is cylindrical and it is bilaterally symmetrical. Okay. So the group of these nematodes will cause elephantiasis. Example, ascaris. Next, annelids. Annelids, example, leech, earthworm. They have true body cavity inside it. Next, arthropoda includes all insects, The circulatory system is open here. So I have already told that circulatory system, there are two types, open and closed. Open means you don't have any blood vessels. But in case of closed circulatory system, it has blood vessels, heart, arteries, veins, everything. So example are, see, butterfly, all the insects, ant, mosquito, beetle, everything. Next, mollusk. Mollusks are bilaterally symmetrical. It can divide it into two equal halves. Then triploblastic. Triploblastic means three layers of embryo. Then they have muscular food for their locomotion. Next, Echinodermata. Echinodermata or triploblastic. So they have three layers of embryo, and also the important character in Echinodermata is that they have hard calcium carbonate. Their skeletons will be very hard because of the presence of calcium carbonate. Example, starfish. Next, vertebrates, we have many phylum here. First one is Pisces, fish. Okay, the body is made up of scales. Okay, they have muscular tail, which is used in movement. Next one is amphibians. Amphibians are cold-blooded animals, and they can live both in land and in water. And their respiration is by lungs and gulls. Okay, the heart is three-chambered heart. Okay, reptiles. So they are three-chambered heart. Okay, they reproduce by eggs, laying eggs. So reptiles, example, snake, turtles, lizard, crocodiles. Okay, tortoise, alligators, chameleon, everything. Next, birds. They are warm-blooded animals. Okay, their forelimb is modified into feathers for flying. Okay, respiration is by lungs. And their heart is four-chambered. Next, last one is mammals. Mammals are warm-blooded animals with four-chambered heart. The skin is covered by hair, sweat glands, and oil glands. So these are present just to maintain the body temperature. Mammals give birth to young ones. They won't lay eggs. Okay. So in case of molecular basis of inheritance, what I have seen, first thing is DNA is genetic DNA. So what are the four requirements of DNA? So that is they must carry information from one generation to another. They must replicate. And they have the property of mutation, then gene expression. Okay, this is the structure of DNA. It has my major and minor group, and it has sugar backbones. So there are two things present: nucleotide and nucleoside. Nucleoside are sugar and nitrogenous base, which is combined with glycosidic bond. Nucleotide is phosphate, sugar, and base. Three components are present in nucleotide. Only two components are present in nucleoside. Purines, pyrimidines. So purines involve adenine and guanine. Pyrimidines, it includes cytosine, uracil, and thymine. So this is the bond formation in DNA. Okay, so these are the comparison of 3 forms of DNA, B, A and Z. In that, B and A are right-handed helix and Z DNA is left-handed helix. So this is the first experiment to demonstrate that DNA is a genetic material. So in that, in first mice, they are injecting the rough strain. Rough strain is very less infective. Smooth strain are more infective. So they are making infected with rough strain first. Then after that, they are adding smooth strain in the second mice. So the mice which is infected with rough strain lives. So it is alive. But the mice which is infected with smooth strain, it gets infected and dies. Then third mouse, which is treated with heat-treated smooth strain. So when the smooth strain is heat-treated, it loses its property of infecting. Okay, so heat-treated smooth strain is added. So after that, sometimes the mouse seemed to be alive. In the fourth category, what he did is heat-treated S strain plus R strain. Both they combine, they are mixed and they are injected into mice. After some time, the mice get infection and it dies. Okay, so this is the second experiment, Avery, Macleod and Mechatic experiment. So what he did is, so he took the heat-killed S strains. So he took the S strains of Streptococcus pneumoniae and he treated with heat. Then he take the heat-treated S cells in a test tube. Then in the second step, he separated, he divided into 3 equal parts. So in the first test tube, the enzyme protease is added. The second test tube, RNase is added. The third test tube, DNase is added. When protease is added, there won't be any protein in this test tube. If RNA is added, there won't be any RNA. If DNA is added, no DNA is present. In the third step, he is transferring the contents into a conical flask. Then he adding R cells in each conical flask. Then after checking the growth of the organism, in first 2 conical flasks, there is growth. So what it indicates that the transformation has been occurred. So in case of R cells in third conical flask, there are no S cells. So what it indicates is no transformation occurs. So finally, they concluded that DNA is a genetic material. If there is no DNA here in this conical flask, no transformation occurs. So thus, DNA is the transformation material. Okay, this is the experiment. Next experiment is Hershey and Chase experiment. Hershey and Chase used radioactive substances. So they use 2 radioactive substances. One is radioactive sulfur and radioactive phosphorus. In first experiment, so he added radioactive substance into the media. That radioactive sulfur will combine only with the outer protein coat. And when it is injected with bacteria, usually the protein coat won't enter inside. Only the DNA will enter inside. And after seeing the bacterial cells through microscope, there is no radioactive cells entered. Okay, in case of phosphorus, radioactive phosphorus, the phosphorus directly labels the DNA. Then after infection, the DNA enters into the bacteria. So if you are seeing the bacteria through microscope, they have seen the labeled DNA inside it. So they found that proteins are not genetic material. DNA is the genetic material. Okay, so these are the persons, a measure who identified DNA. So he identified the DNA first from the pus cells. Okay, he named it as nucleon. Next person is Altman. Altman named it as nucleic acid. And the person and the two scientists, Watson and Crick, gave the structure of DNA. And they got Nobel Prize in 1962. Okay, next, purine pyrimidine we have already seen. This is Chargaff's rule. Concentration of purine is equal to concentration of pyrimidine. Okay, adenine, how much of concentration of adenine will be equal to the concentration of thymine. Likewise, concentration of guanine will be equal to concentration of cytosine. Okay, they have given a sum. If adenine is 30%, what will be the thymine? And what will be the other bases? So if adenine is 30%, thymine will also be 30%. So everything will be adding to form a 100%. So 60%, 100. So 100 minus 60, 40. So G and C is combined to form 40. Then if it is divided by 2, 20 and 20. Next, bonds in DNA. What are the bonds present in DNA? Hydrogen bonds, phosphodiester bond. Okay, DNA is a negative charge. Okay, so it can undergo denaturation and renaturation process. When DNA is treated with heat, the strands will separate. So that is called denaturation. And when it is cooled, it will renature again. Next, RNA. So some scientists like Frankel and Singer described that RNA is also a genetic material. So they did an experiment with that. They used the experiment along with tobacco leaf and tobacco mosaic virus. So what they did is they took two types of tobacco mosaic virus. In that, they removed the protein from RNA strand and they exchanged the proteins with RNA strands of the two forms of TMV. Okay. Then they allowed it to infect with the tobacco leaf. After the infection, when they were seeing the progeny, the infection in the microscope, they have seen the original protein form of the original RNA. So what they are concluding is that the RNA is the genetic material. Okay, what are the types of RNA? tRNA, mRNA, rRNA. So everything tRNA, mRNA, rRNA. 3 RNAs will be helps in the process of protein synthesis. Okay. So next, some other RNAs are there which are used in laboratory research purpose. So these are the differences between DNA and RNA. They have a deoxyribose sugar and ribose sugar. And in DNA, thymine is present, uracil is not present. And in RNA, uracil is present, thymine is not present. So can you understand?